Everyone, my name is Phillip Oelbaum. I'm just completed my PhD in the Welch Lab at the University of Toronto, and one day I'll be starting a postdoc in the Clare Lab at York. So my affiliation is in flux, but this is a chapter for my PhD, and I tried to sort of tune this towards the Ontario Bat Network and why you guys should care about a community of bats in northern Belize. So fundamentally, when we're thinking about studies in ecology, populations, communities, ecosystems, the unit that we're looking at is the individual. Individuals comprise these populations, and you know, as we go up through these levels of organization, we really need to be thinking more about the individual, because individuals do vary in ways that we measure all the time. They can vary in age, class, sex, and reproductive status, things that we get when we're catching bats. Body size, morphological characteristics, forearm, mass, things like this, coloration even. There are also evidence to suggest that bats have different personalities or behavioral phenotypes, and a lot of this work has come from Jerry Carter's lab on vampire bats, but also there's some upcoming work from Caleb Ryan and Rodder's lab that is very interesting, so keep a lookout for that. And then the thing that I was interested in here was, do individuals demonstrate these preferences? So two types of individual specialization that I was thinking about here is both dietary preferences and sort of foraging, spatial, foraging ecological preferences. So selective use of prey items and selective use of space. And this is widely described across bat taxonomy in these snapshot studies. So here's just a few examples. Canadian bat, Antrozoa pallidus, found in BC. But in southwestern United States, different individuals will prefer different food items. So Johnson Fenn showed that some are scorpion specialists, some are centipede specialists, things like that. Io in China and Southeast Asia. There's recently a couple of studies showing this from molecular data. And then also similar time frame, Myotis anteri in Europe has a very similar effect. When we're thinking about use of space in terms of foraging preferences, this is a study from Finland on barbastal bats. And this is what they were looking at originally was a cohort level effect of males and females foraging in different areas. But when they sort of zoomed in, there are these individuals that are foraging in sort of specified areas. This is a study from the Oelbaum lab in Israel, and this is two different species of Myotis. One that's social foraging on fish over water, over a broad area. They've got this sort of ephemeral resource on the right. And then on the left are individuals sort of tracking out from a roost on their own, consistently to their own foraging locations. These studies are all done using GPS tags. To move away from vespers, just to show you this one really extreme example, this is Leptonicter zerguene. They're a phyllostomid from Mexico and the southwestern U.S., the tequila bat. And these are GPS tracks. One of these bats, the one where it says first night there, flew, I think it was over 150 kilometers in a night, out to its foraging location and back to its roost. And it did this several nights in a row. And all these bats have their own sort of preferred little areas. So I wanted to know, when we're trying to look more broadly, these are all very targeted studies, sort of one-offs, snapshots at a particular time. How would we go about monitoring for individual specialization in a broad community, in a diverse community? How widespread is this individual specialization in bats? Because I found, I think 10 examples across a lot of different taxa. But looking at one place where there's a lot of species co-occurring, how do we look at this? And I turned to stable isotope analysis, which is a technique that I used in my master's. I don't have a pointer, do I? Let me switch this. There we go, these are pointers. All right, so just to sort of walk you through this quickly, carbon down here on the x-axis, delta 13C, ratio of heavy to light carbon in animal tissues is preserved from the original source of carbon in the environment. So you get some level of spatial foraging information. If your bat's foraging on the grassland, it would have this C4 signature. If it's feeding in a forest, as the C3 signature. Marine systems and aquatic systems would be down here, very different. There's also a canopy effect within carbon, but I don't have time. And then nitrogen here on the y-axis, that's delta 15N. That increases with trophic levels. So our herbivores have a lower nitrogen ratio, lower nitrogen, the omnivores have lower nitrogen than carnivores. And then using these together, we're able to map on these isotopic niches. So these are 2 dimensions of what is, the n-dimensional ecological niche. Animals exist in a lot of different dimensions. But this confers both dietary information, so what the bats are eating, and some information about spatial foraging. So looking at this in a bat context, what does this look like? This was from a 2019 study I did at Lamb, Belize. And this was our prediction figure of, we can put these bats into these broad trophic guilds. Where do we expect them to shake out? So our fruit-eating bats probably have the lowest nitrogen. Our nectar-feeding bats, we know they tend to take a bit more insects than fruit-feeding bats, so stack them up. And then generally insectivores and carnivorous bats get this sort of trophic structure. So carnivores at the top. And then fish-eating bats over here with this aquatic signature, and our vampire bats who are thinking of feeding on livestock, so over here. When we put this into, get the real data in here, that is pretty close to what's happening. There's a few weirdos. This is an insect-feeding bat that forages directly over the surface of the water. This insect-feeding bat probably forages more over open space. But generally, yeah, this is consistent with our expectations. So now that you guys have sort of sense of stable isotope analysis, what I was interested in doing here was like, say this big ellipse here, this is the isotopic niche for Desmodus rotundus, the common vampire bat. I wanted to know if this individual is always different from this individual. If we have marked individuals, trying to see how similar they are to themselves over time, as opposed to just sort of randomly distributed within this population's niche. And the reason you guys should care is because this happens in bats found in Ontario too. So this was a study by Paul Cryan. This looking at big crown bats in Colorado, I believe, and they demonstrate this individual isotopic specialization. It's tough to do though, because you really need to be able to go back to the same populations, you marked individuals, and consistent sampling efforts and procedures over time in order to be able to sort of look at this. So one of the really good place to do this was Lamini. And we used pit tags in order to do it. And this was an enormous amount of effort. Every bat that left the classroom or sort of central processing location got a pit tag. We ended up, I think we're close to about 3,000 animals tagged now with about 16% recapture year over year. And this has been going on since 2021. So we have a good sample base for a lot of these species. However, it's just sort of a scattering of samples for each species. So I'm going to sort of pare it down a bit. So looking at these populations of hundreds of individuals, I wanted to know how these individual preferences are driving resource partitioning. Is it that, I'm this Glossophaga here that's feeding on banana flowers, but my friend is a Glossophaga that eats mostly insects, and that way we're not competing for resources. It reduces intra-specific competition. So using that data from the 2019 paper I showed you earlier, as well as the data from recaptured individuals between 2021 and 2024, I want to compare the isotopic position for individuals within these populations. So I hypothesized that this individual specialization is widespread among the tropical bats, stable over time, and that individual specialization will contribute to this niche breadth that we see. So in terms of our largest niche breadth, those vampire bats, it's individual specialization, different corners of that niche that's driving that size up. We did 2 different statistical analyses. So we did a permutation analysis, essentially to generate p-value that answers this question: am I more similar to myself than I am to others in 2 dimensions? And then we also used mixed effects models with intra-class coefficients for each dimension independently, so carbon and nitrogen. And if you see these values closer to 1, that means that individuals are highly repeatable over time, closer to 0, low individual repeatability. So just to, I don't know how much time I have, but I can sort of rush through the actual meat of the results here. These are 3 species of fruit bats, all of them in the genus Artivius. And at the top here you can see our permutation analysis came back, yep, you have, you're more similar to yourself than you are to others within the same population. All these bats are sort of generally nomadic, so that's not necessarily a surprise. They're probably coming from different areas and recapturing the same places. But you can see sort of the colorful points here. These are the recaptures, and black are 2019 data. And it's looking in single dimensions. Artibius liberatus and Artibius germescensis here are generally evenly distributed in both. So they vary equally in nitrogen, vary equally in carbon. But Artibius intermedius, this other species over here, is very low ICC for carbon, suggesting that they might be foraging sort of more broadly or coming in from various different systems, as opposed to, or relatively the same system rather, as opposed to that. But nitrogen is quite high, so this green individual probably has a very low insect consumption as compared to this blue or yellow individual up here. So these are two species now where our permutation analysis came back and showed that individuals are not more closely to themselves than to others. However, when you look at our ICC values, single dimensions again, nitrogen, there's really high ICC. So for Glossophaga, this is the case again, where this yellow individual has consistently lower insect consumption than this red individual than this blue individual. And Molossus nigricans is an insect-eating bat. There's actually a dietary study that showed that individuals do have dietary preferences from Fenton et al., where, say for example, this blue individual is probably feeding more Lepidoptera consistently, while this one is more Coleoptera, and things of that nature. So that's actually quite supported by existing literature. So the last example I want to show is kind of this quite cool. So this on the left here is Corollia perspicillata. They are the generalist fruit bats, as far as we know. Basically, trash bat in the Neotropics. You find them everywhere. And in our system, in the original study and currently, they had the smallest isotopic niche breadth. So typically, when you see something with a small niche breadth, you say, okay, that's a specialist, or we would think of that as a specialist. And then over here on the right is Desmodus rotundus, vampire bat. They eat blood. They pretty much only eat blood. But they had the broadest isotopic niche of anything by magnitudes. So you have a dietary specialist with a really broad niche suggesting they're a generalist bat with a very tight isotopic niche suggesting they're a specialist. And that's a case where this sort of individual generalism is collapsing Corollia's niche breadth down to a mean. So when you're eating, your isotopes are sort of averaging out your tissue. All of these bats are sort of equal in the way that they're foraging, at least in terms of what the isotopes are telling me. And they are this sort of generalist of generalist where equal parts throughout the population. For a Desmodus, we know they have these huge social relationships where they share resources. But here there's very strong evidence, both permutation and from our mixed effects models, to suggest that individuals are specializing in something in particular. So just to show you visually in terms of the isotopic niche breadth for both of these, yeah, it's quite striking. So just to wrap up here, go back a little. So permutation analysis, we found that there was evidence to suggest that 11 of the 15 species we considered, including both of the species of Vespers, demonstrate this isotopic specialization. When we added in the mixed effects model data, it's 14 and 15. So this is really widespread in bats. It seems like it's the exception, not the rule, that bats are sort of this generalist, right? So Desmodus rotundus, again, population level generalist, but strong evidence suggests individual specialization, and Corollia the contrary. So we found that individual specialization is widespread in tropical bats. I hope that I've convinced you that this is a good, interesting thing. Consider the role of the individual when you're doing ecological studies. This is an enormous collaborative effort from a lot of different people. The pit taggers are, we work all night, and I can't stress that enough. And I'm happy to take any questions.
So for the vampires, do you think it could be like down a level? Like even though they just eat blood, they probably eat blood from different things, not just cattle, and maybe some, cats, or something like that. Could that contribute to it?
There was a study by Dan Becker that from the same system where they tried to use mixed effect, single isotope mixing models to sort of figure out what they're actually eating, and sampled blood from a bunch of different cows, from a bunch of different farms, and couldn't fully reconstruct diet. So there's something that we're missing there. The interesting thing with that is it's perhaps a cohort level effect where there are small groups of vampire bats that are more similar to each other, but just because our sample size, I think we had 6 individual vampire bats there, we're just not picking that up. So I'm interested to go back and continue this to see if we can start to tease that out. Still good, okay. Liam?
Neat stuff. For the individual specialization, individuality side of things, I'm wondering, do you have to account for sample size? In the sense, if you only sample 3 individuals, they could look quite different. But if you're making 10, 50, 1,000 individuals, you're going to start to get a cloud. So how do you factor that into the degree of specialization of the population level?
Yeah, so the question was how to account for sample size. And to be totally honest, for a lot of these, we just can't right now. We just don't have the sample base. I think like a 10-year retrospective on this would probably yield the answers to that, but these are the samples that were available from bats that we captured 2 or more times. So it could be that some of this strong signal that we're seeing will start to fade out. It could be that we're seeing cohort level effects like males and females are different, or certain age classes that we just don't see yet. But with the evidence provided right now, this is as good as I can get it. Good. Okay. Thank you.
Thank you so much. Our next talk is going to be by Aidan Lueders, Terrestrial Locomotion Bats, Associations of Gait Characteristics with Body Size and